So that, Dr. Sara, you can just proceed it.
Hello, everyone. Is my voice clear?
Yeah, it's fine.
Okay, so good afternoon to all of you. And today I am going to present that how microorganisms, especially microalgae, they produce the biofuels. So I'm Dr. Sara Junaid, as the Suman has already introduced me. So just move towards the presentation, and now I'm going to share my screen. Wait a second. Okay, is it visible to all of you?
Okay, it's now.
It's visible right now?
Yeah.
Okay. So I am Dr. Sara Junaid from the Department of Microbiology and Molecular Genetics. And here are some acknowledgements that these organizations and my supervisor who involved in the completion of my PhD research work. So come towards the multifaceted biofuel production from the microalgae. So I will discuss the introduction, sampling and isolation and characterization, biodiesel production, biohydrogen production, bioethanol, and then we will conclude all the results. Now, come towards the biofuels. What is biofuel? Biofuels are a renewable energy source from which we can obtain from any type of the biomass, which can be the living biomass, and biofuels are capable to replace the transport fuels which are we're using right now. So there are different types of the biofuels which we can obtain from the biomass, including biohydrogen, biomethane, bioethanol, and biodiesel. However, I will discuss with you regarding with the biodiesel, bioethanol, and bioethanol and hydrogen. First of all, come towards the generations. That what does generations mean? First of all, when the scientists thought that we must obtain the biofuels from any living source, so they moved towards the different plant sources. In the first generation, when they obtained the biofuels, so those biofuels were from those crops which were used as a source of the food as well. So scientists thought that why we are using those plants as a source of biofuels which we are already having as a food. So that's why they move towards the other generation, which is known as the second generation of biofuels, and the biofuel was obtained from the known food crops. Now come towards the third generation, from which we can obtain the biofuel from the microalgae and the cyanobacteria. So I will discuss regarding with the third generation of biofuels. There is also the fourth generation of the biofuels, in which we genetically modified the organisms and we obtained the different types of the biofuels from the microorganisms. What is cyanobacteria and algae? So these are the photosynthetic microorganisms, and the main differentiating point among them is cyanobacteria are prokaryotes. However, microalgae are the eukaryotic in nature. And cyanobacteria is also known as blue-green algae. It has the different pigments known as the phycobilin and the phycocyanin, due to which the cyanobacteria appears blue-green in color. However, both of the creatures and the microorganisms have the chlorophyll in their cells. So this is the pictorial representation that this is a microbial cell, and how we can obtain the biohydrogen, bioethanol, and biodiesel from these microorganisms when they use the different nutrients from the environment, and they can utilize all these nutrients and they gave us the different types of biofuels. So aims and objectives of this study are that we screen the cyanobacteria and microalgae from the unexplored areas of the Pakistan, and we extract and measure the total lipid content as well as the biodiesel yield and the chemical composition of the biodiesel. And the second main point was the direct ethanol production was obtained from these microorganisms, and as well as the hydrogen production as a source of biofuels. The sampling was done from the different areas of the Pakistan. So here I have mentioned the 6 different areas, including the northern areas and as well as the Punjab areas. So when the samples were transported to the lab, they were further processed for the isolation and the purification process. And there are the different types of these microorganisms. Some are known as the unicellular and some are known as the filamentous microorganisms. So unicellular microorganisms or microalgae, they are similar in appearance with reference to the unicellular bacteria. So we just took the sample and we just processed it in the BG11 media. So this is the specific media which we use for the growth of these green creatures. So BG means blue green. And then we examine under the microscope, and after that we have to isolate as we follow the same protocol which we use for the simple bacterial cells. And then we enriched these microorganisms in a BG11 media. So here in a flask, in the last we obtain the pure culture. Now come towards the filamentous cyanobacteria, how we separate and how we purify them. We place the sample into the lab, we put it into the BG11 media, examine under the microscope, and then we place a filament on an agar plate, solid agar plate. And then after the growth of these microorganisms on the solid plate, we shift this agar sphere into the another plate. So by this passaging method, we purify the filamentous cyanobacterial strains, and we then observe under the microscope when they are isolated and purified. Then we shift it into the BG11 media again, and then there we can obtain the enriched. So into the 65 different strains from the different regions of the Pakistan, 33 were unicellular microalgae, and among the cyanobacteria, further 13 were 13 were under the heading of unicellular cyanobacteria, and 19 were under the filamentous cyanobacteria. Now come towards the identification method. So different identification methods were obtained for their identification. These were morphological identification and as well as the genetic identification. Now come towards the morphological identification. For the identification of the cyanobacteria, I used the different protocols for their identification. However, for the microalgae, I used these different key terms for the microalgae identification. Now come towards the genetic identification. Here is a differentiating point among the genetic identification of the cyanobacteria and microalgae, that for the cyanobacteria, we focus towards the 16S ribosomal RNA sequencing. However, for the microalgae, we use the 18S ribosomal RNA sequencing technique for their identification. When we obtain the specific microorganisms and after their purification, we send these microorganisms for the sequencing, and after obtaining these sequencing, we just compare it with the NCBI, and then we perform the phylogenetic analysis. And I have performed the phylogenetic analysis of all the 65 strains which I used it during my PhD research work. So here is a pictorial representation in the form of the graphs of how much filamentous cyanobacteria was obtained. So here is how much unicellular cyanobacteria was obtained. So the highest number was among of the Cyanococcus among the unicellular cyanobacteria. And among the unicellular microalgae, the highest number of Chlorococcum strains was obtained among all species. So step by step, I will discuss each type of the biofuels with you. First of all, I will discuss with regarding the biodiesel production. So what is biodiesel and how we obtain it? Biodiesel is actually the monoalkyl esters of the long chain fatty acids which we obtain from the biomass, and these biodiesel can be used in the vehicle engine. So you are very well aware about the term of the biodiesel. And when we obtain this diesel from the microorganisms or from any living organism, then this is known as the biodiesel. So what happens that microalgae or cyanobacteria, which are naturally growing in the environment, so they obtain the water and the sunlight as the source of the energy, and they also utilize the carbon dioxide for their growth. So whenever these creatures or microorganisms, they grow, then according to their growth, the lipid particles or lipid bodies, they also develop within the cells. So these lipid particles are basically used as the source of the biodiesel for us. So we harvest the cells, and then we extract the lipids from them, and then we isolate it, and then we compare it, and we obtain the biodiesel. So here are the different steps that we have to select the microalgae strain specific, and then we have to harvest the biomass, and after that we have to extract lipid, and for those lipids we do the process of transesterification, and after the transesterification we obtain the biodiesel. So what is transesterification? Basically, the triglycerides which are already present in the cells, in the microbial cells, so we treat it with the methanol. So in the presence of the methanol and the catalyst, this triglyceride will be broken down and it will be converted into the fatty acid esters. And these fatty acid esters are used as the source of the biodiesel. And we can also obtain as a waste of the glycerine. And we can use this glycerine for the remaining other purposes. If we are discussing about the biodiesel, so how we can forget about the word of lipid production? So firstly, we will have a greater focus towards the lipid production. So here are the summarization of my results which I performed. I used the cyanobacteria and microalgal strains in the different BG11 conditions to obtain the different amount of the lipids from these microorganisms. So I used the different percentages of the sodium nitrate in the media, which is the basic source for their growth. So I used 0 to 2% of the sodium nitrate in the BG11 media, and as well as I also used two different growth conditions. Some same strain was grown in the lab and as well as in the natural environment. And then we compared the lipid production among these all different conditions. So highlighted regions is showing the highest results which we obtained from these microorganisms. So in the case of the filamentous cyanobacteria, the maximum results were shown by the oscillatoria. However, leptolingia also shown the best results in the case of the 1.5% sodium nitrate. Here is the results of the unicellular cyanobacteria, and same the highlighted regions are showing the highest results which was obtained from these different microorganisms. And here are the results for the unicellular microalgal strains, and all the lipid amount was compared among these microorganisms. Now come towards the summary that among all the filamentous cyanobacteria strains, the oscillatoria species showed the lipid production up to the 70% in 0.5% sodium nitrate. However, unicellular cyanobacteria Cyanococcus showed up to the 50% lipid, and microalgae showed up to the 79% lipid production in presence of 1% sodium nitrate. So these microorganisms were considered as best lipid producers. When we obtained the lipid, so come towards the biodiesel productivity. My slides are visible to you?
Yes, it's visible.
Okay. I thought that it is not visible. Now come towards the biodiesel productivity. So biodiesel productivity was measured in the mgs per liter per day, and the highest values and highest amount of the biodiesel have been highlighted in these tables, and you can see that maximum biodiesel productivity was obtained in the oscillatoria strain and in the leptolingia strains. And with reference to the unicellular cyanobacteria, highest biodiesel productivity was obtained, and same different conditions were used for the sodium nitrate to observe that how much biodiesel can be obtained from these microorganisms. Here are the results of the unicellular microalgae, and summary of the biodiesel productivity from these microorganisms is that among the filamentous cyanobacteria, leptolingia showed the maximum result. Unicellular cyanobacteria showed the among the unicellular Cyanococcus showed the maximum result, and among microalgal strains, Chlorococcum species showed the maximum result to produce the biodiesel from these mini creatures. Now come towards the FAME. FAME is known as fatty acid methyl esters. As I told you that during the transesterification, we obtain the methyl esters, and these are actually known as the biodiesel. So I detected these fatty acids from these microorganisms, and I used 14 different types of the fatty acids to detect among these microorganisms. And among these 14 types of saturated fatty acid types, 9 were saturated. However, 3 were monounsaturated and 2 were polyunsaturated fatty acids. So I detected that how much amount of saturated or unsaturated fatty acids were present in these microorganisms. So here are the results of the GCMS, which are showing the peaks of the different carbon number among the different microorganisms. And now come towards the filamentous cyanobacterial results, and it is showing the relative quantity of the fatty acids in the lipids, and all the 14 fatty acids which I used to detect in these microorganisms. So here are the results, and the red highlighted region is showing the maximum result and among the saturated fatty acids, and green highlighted region is showing the maximum result among the unsaturated fatty acids. So you can see here the results. Here are the results of the unicellular cyanobacteria. Same with reference to the microalgae. The maximum results have been shown. And here is the summary of the results, that among the filamentous cyanobacteria, the highest number of C18 3 fatty acids were obtained. However, in case of 3220 fatty acid was not detected. So this is the FAME and fatty acid methyl acid. These are the types. See, when we show any number with the carbon, so these are showing the FAME. So here we compared the FAME analysis among these microorganisms, and so you can see that C80 and 18 were not detected in the cyanobacteria. However, among the unicellular microalgal strains, the Dictyochlorobsis it showed, and Botryophocis showed 13 types of fatty acids. Because I detected the 14 types, and among all the 14 types, 13 fatty acid types were present in these 2 types of microalgal strains. Now come towards the summary of the fatty acids. Mono and polyunsaturated fatty acids, they were higher in number in the cyanobacteria and microalgae both. However, the ratio between the saturated and unsaturated fatty acids, it was higher on the unicellular cyanobacteria. So if we say that cyanobacteria is prokaryote and microalgae is the eukaryote, and on the basis of their fatty acid production, there is also a difference, that unicellular cyanobacteria have the more amount of the saturated fatty acid in comparison to the unsaturated fatty acid. Now come towards the another techniques, which is known as the fluorimetry, and this technique was used for the detection of the lipids within the cell, and it was measured on the 2 bases. We measured it on the quantitative basis and as well as the qualitative basis. So first of all, I will discuss regarding with the quantitative analysis of these lipids via the fluorimetry. And for that purpose, I used the 2 fluorescent dyes known as the body P and as well as the Nile red. And via using these dyes, I detected the higher RFU. RFU means relative frequency units, which I detected from these microorganisms by using the fluorescence of these dyes. Here are the results showing the maximized results, and all the results are shown in the form of the graphs for the different types of the cyanobacteria and the microalgal strains. So here is the summary of those graphs, that among the filamentous cyanobacteria, oscillatoria showed the highest RFU value with the body P dye, and the 30 RFU value with the while using the Nile red dye. And same are the results showing with the unicellular cyanobacteria and the unicellular microalgae. So in short, or in summary, if we will say that body P dye showed the better affinity for the intracellular microlipids. Now come towards the localization of the lipids, that how we can detect the lipids, that where they are present within the cells. So it can be detected by the 2 different techniques. One is known as the Sudan black staining, and second one is known as the CLSM. CLSM is an abbreviation of confocal laser